Assalamu alaikum. So now that we have talked about enzymes and their features and what they do exactly, in this lecture, let's go into classification of enzymes. Okay. So now, enzymes usually, when they are named, there is a special system on how to name enzymes that scientists have agreed on. And usually this system, basically the name ends with a suffix "ase." Okay, so and usually it describes what the enzyme does to what substrate. Okay, so we can say something like ATPase, and this means, again, this is an enzyme and it breaks up or breaks down ATP. We can say ATP synthase, and what it does is that it synthesizes ATP, and so on. Okay, so we'll talk about the different naming systems right now. Now, sometimes before scientists agreed on the system, they used to name or to give enzymes common names. So, for example, you have the official name of someone like Muhammad, I don't know, Abdullah, and so on. That's the official name, and there are names that are common. So, for example, you can name someone Abu Shakush, okay, and so on. So, an example is trypsin. Trypsin is a protease. Okay, it should have a specific name, but its common name is trypsin. Now, ask your parents about Abu Shakush. In the 90s, he was really famous. Okay, so, and what he used to do was he would use a hammer to kill someone in a store, dukkane, whatever, and steal the money. And he did that several times before he got caught. Ask your parents. So, enzymes are divided into 6 major groups. And these are the groups: Oxidoreductases, the largest group. We have Transferases, Hydrolases, Lyases, Isomerases, and Ligases. Okay, so let's talk about these groups and what they do exactly, and how we can look at a reaction and decide on the name. So let's start with the first class, Oxidoreductases. Now, this class contains the largest group of enzymes, and they have different functions, but what they do is basically the same thing. They catalyze oxidation reduction reactions. Okay, so you have a transfer of hydrogen atoms or electrons from one molecule to the other. They have been divided even further into 4 subclasses, and these are Dehydrogenases, Oxidases, Peroxidases, and Oxygenases. So this is what we will do. I'll talk about each one of these subclasses. I'll talk about each class. Okay, I'll give you an example, or more than one example of a reaction. And in the future, if you see a reaction, I want you to practice by designating the class that the enzyme belongs to. So in the exam, for example, I'll give you a reaction, and you have to conclude what the class is that this enzyme belongs to. So let's talk about the first subclass. So dehydrogenases transfer electrons in the form of hydride ions or hydrogen atoms using an electron transferring coenzyme. And this coenzyme is usually NAD or NADH or FADH2 or FAD. So we have to examine, first, what is a hydride ion? So a hydride ion is basically a hydrogen with a negative charge. It's the negative ion of a hydrogen with reducing properties. So what it does is that in this example, in this first example, we have lactate dehydrogenase. And what it does is that it takes a hydride ion from lactate and it gives it to NAD. This is an electron carrier, okay? And the products are pyruvate plus NADH plus H. So the hydride ion is split into two parts. First, you have the hydrogen combines with NAD, forming NADH, and you have the release of a proton. Now, pay attention that the reactions that I have chosen as examples are reactions that you will study in the future in the metabolism course. So these are really important reactions that you should know. In the exam, I will not ask you specifically about a certain enzyme by name and tell you or ask you to what class it belongs. From the name, you should be able to tell. That's one. Two, I'm going to give you an example of a reaction regardless of what the enzyme is and ask you to what class this enzyme belongs to. So here's another example. You have alcohol dehydrogenase. What it does is that it takes ethanol, it takes a hydride ion from ethanol and it gives it to NADH, producing NADH plus H plus the oxidized form or an oxidized product, which is acetaldehyde. Okay, now notice that the reaction, both reactions are reversible. So the same enzyme catalyzes the opposite or the reverse reaction. So what it does is that it takes the hydride ion from NADH plus H, the proton, and it gives it to pyruvate, producing the reduced form or the reduced molecule lactate. Same thing with alcohol dehydrogenase. The second subclass is oxidases, and these enzymes transfer hydrogen from a substrate to molecular oxygen, okay, producing hydrogen peroxide as a byproduct. So this is the reaction right here. So you have glucose plus oxygen. Now you have the transfer of hydrogens taken from glucose to oxygen, producing hydrogen peroxide, okay, and the oxidized molecule. So the electrons are taken in the form of hydrogen from this group right here. Now, so this is how you can differentiate this subclass or enzymes that belong to this subclass. That is, you have the substrate as oxygen and the product hydrogen peroxide. In the dehydrogenase reaction, the previous enzyme that we talked about, one of the substrates should be an oxygen carrier, that is either NAD, NADH, or FAD, FADH2. Okay. Now notice that these two are isomers, right? They differ in the position orientation of the hydroxyl group that is bound to the anomeric carbon, to carbon number 1 of glucose. It's either below the ring, alpha, looking like a fish below, or above the ring, that is beta or bait. Bait is always above the ground. So it's above the ring. Now the third subclass is peroxidases. And these enzymes catalyze oxidation of a substrate by hydrogen peroxide. Okay, so as a substrate, you have hydrogen peroxide. And a great example is an enzyme known as glutathione peroxidase. This is glutathione. Glutathione is a reducing agent, so it it reduces other molecules and it gets oxidized. So it is a reducing agent. Glutathione is composed of 3 amino acids, and these are glutamate, glutamic acid, so you have a hydroxyl group, negative charge, cysteine with a sulfur group right here, a thiol group right here, and then glycine. Okay, so these are the 3 amino acids. And what happens is that glutathione, you have 2 molecules of glutathione that get oxidized, forming a disulfide bond right here. And you have another molecule that gets reduced, which is hydrogen peroxide, forming water and oxygen. So what it does, it is an antioxidant enzyme and removing harmful hydrogen peroxide from cells. Now, the reverse reaction also takes place by taking electrons from an electron carrier known as NADPH. So this NADPH gets oxidized, and the diglutathione molecule, the oxidized glutathione molecule designated as GSSG, because you have a disulfide bond, gets reduced, forming the reduced glutathione form that can remove another oxidizing agent. Now the fourth subclass is oxygenases. So be careful, you need to differentiate between oxidases and oxygenases. And what oxygenases do is that they oxidize substrate by molecular oxygen. So as a substrate, you have oxygen just like oxidases, right? Except that the product is different. So right here what happens is that the substrate is oxidized by molecular oxygen, producing oxygen, introducing oxygen into the substrate. So the reduced product is water. Rather than hydrogen peroxide like oxidases, water is the product. And the other thing is that these oxygenases can be monooxygenases or dioxygenases. Okay, so what's the difference? The difference is how many oxygens are transferred to the substrate. If one of them is transferred, then they're known as monooxygenases. If both of them are transferred, then they are known as deoxygenases. So notice here in this reaction right here, you have one oxygen transferred to the substrate, producing the reduced form, the oxidized form, and you have the formation of water. Right here in this reaction, reaction by catalyzed by dioxygenases, you have transfer of oxygen to both of them to the same substrate. Okay. So, same thing here. These are deoxygenases, and this is monooxygenase. So, that's the first class, Oxidoreductases. The second class is Transferases. And what they do is that they transfer a functional group, and this functional group is either carbon, nitrogen, phosphorus, or sulfur from one substrate to another. Now, a great example of transferases is kinases. And what they do is that they transfer a phosphate from a phosphate carrier like ATP. So ATP has a functional group of phosphorus, and it gets transferred to a substrate, to another molecule. For example, you have fructose 6-phosphate, and one phosphate is taken from ATP, producing ADP, the adenosine diphosphate, from adenosine triphosphate, and it's conjugated or attached to fructose 6-phosphate. Now you have a product known as fructose 1,6-bisphosphate. So here's a question. So what's the difference between saying diphosphate and bisphosphate, since both of them have two phosphorus or phosphate molecules? The difference is, as in adenosine triphosphate or adenosine diphosphate, is that both of these phosphates are linked to each other. So it's known as adenosine diphosphate. Whereas here we have fructose 1,6-bis phosphate. Bis indicates that this molecule contains two phosphates, that is bi, except that the two phosphates are not linked to each other. They exist on two different groups. Another example of transferases is what is known as transaminases. And what these do is something that is very critical. They transfer amino functional groups from one molecule to the other. And usually one of these molecules is an amino acid and the other molecule is a keto acid. Okay, so here's an example right here. You have glutamate and oxaloacetate. Glutamate is the amino acid and oxaloacetate is the keto acid right here, for example. It's a keto acid. And what happens is that you have this amino group taken from the amino acid and it's given to oxaloacetate. The glutamate becomes a keto acid, also known as alpha ketoglutarate. And oxaloacetate, now that it has an amino group, it gets converted to an amino acid. So you have interconversion of amino acids. Glutamate can be used to produce aspartate, and this enzyme is known as aspartate transaminase. Now, notice that the reaction is reversible, meaning that the same enzyme takes amino group from aspartate and it gives it to alpha ketoglutarate. Alpha ketoglutarate becomes glutamate and aspartate becomes oxaloacetate. So here you have amino acid and keto acid, and the products are also amino acid and a keto acid. The third class is hydrolases. Now, hydro means water, and lases, basically, it's breaking up a bond. So what these enzymes do is that they use water to break bonds. Okay, and a great example, or examples, I should say, you have proteases. They degrade proteins, they cleave proteins. Esterases, they remove ester groups. Lipases, they cleave lipids. Glycosidases, they cleave sugars. Phosphatases, they remove phosphate groups. So these are all examples of hydrolases. They use water to break a bond or release a functional group. So yeah, we already talked about proteases, trypsin, chymotrypsin, elastase, and what they do is that they use water to break a peptide bond. Okay, so you have a peptide bond right here. One of the groups that are involved, one of the groups that is involved in the peptide bond gets a hydroxyl group, and the other one gets a hydrogen ion. So we already talked about trypsin, chymotrypsin, elastase, and already talked about these enzymes and how specific they are. So trypsin recognizes lysine or arginine in a peptide and cleaves this peptide at a peptide bond involving lysine and arginine. Chymotrypsin recognizes phenylalanine, tryptophan, and tyrosine. Elastase recognizes a peptide or peptides that contain glycine, alanine, or valine. So now the fourth class is lyases, and what lyases do is that they cleave bonds between carbon and carbon, carbon oxygen, carbon nitrogen, or any other bonds involving carbon and a group, reactive group. And these enzymes do so by elimination. So they produce a product, a molecule having a double bond or a ring, or they can do the opposite. That is, they can add a double bond to a molecule. And there are even subclasses of these lyases, of these enzymes. You have enzymes known as dehydrases. So what they do is that they remove water from a substrate by creating a double bond. An example is enolase. Okay, so what's like phosphoenol pyruvate. So en, by the way, indicates a an ethene double bond. Ol is an alcohol. So right here, phosphoenol pyruvate, you have both. You have a double bond like here between two carbons, and there is a hydroxyl group in this molecule, which is right here. Okay, except that it has a phosphate. Okay, so that's a dehydrase. Another example is decarboxylases. And what these enzymes do is that they remove a carbon in the form of CO2, and that's why they're known as decarboxylases. So they remove CO2 from a molecule, like converting pyruvate to acetaldehyde. And you have synthases as well, and these molecules, they add a small molecule to a double bond, so they remove a double bond. Remember, I said that they either add or they remove double bonds. So you have citrate synthase that converts that combines oxaloacetate to acetyl coenzyme A, or the opposite. They add acetyl coenzyme A to oxaloacetate, forming citrate. So they remove a double bond within a molecule. Another example is an enzyme known as aldolase. And what aldolase does is that it breaks down fructose 1,6-bis phosphate. So what it does is that it removes, or it breaks up, an aldol molecule, a sugar molecule with an aldehyde group, like as in fructose. Okay, so what it does is that it breaks up fructose 1,6-bisphosphate, this molecule right here, into two molecules known as dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, forming and cleaving an aldol molecule. Okay, so right here you have breaking up an aldol molecule, a hydroxyaldehyde, forming another hydroxyaldehyde, that is glyceraldehyde 3-phosphate, as well as dihydroxyacetone phosphate. Another example is enolase. What enolase does is that, again, it forms an enol molecule, a molecule with an ethene, a double bond, and the same carbon is involved in the formation of hydroxyl group as well. That is right here. Now, isomerases, they're really easy. They interconvert isomers. Okay, so an example is phosphoglucoisomerase. Basically converts a glucose molecule into a fructose molecule. In this case, glucose 6-phosphate to fructose 6-phosphate. These two are isomers. Okay, another example, so right here, that's a phosphoglucoisomerase. Okay, so these are two isomers, two molecules. Now, another example is phosphoglycerate mutase, and what it does is that it converts 3-phosphoglycerate to 2-phosphoglycerate. So what it does is that it takes a phosphate group from carbon number 3, and it puts it at carbon number 2. So these two are isomers as well. So that's what isomerases do. And usually reactions catalyzed by isomerases do not require energy. The final class is ligases. And what ligases do is that they glue molecules to each other. So they join carbon with another carbon, carbon with an oxygen, carbon with an nitrogen or sulfur, or a halogen molecule. Now, usually, since we are talking about conjugating, attaching a molecule to another, this reaction usually requires energy in the form of ATP. An example is pyruvate carboxylase, that is attachment of a carbon dioxide in the form of carbonic acid, bicarbonate ion to pyruvate, enlarging this 3-carbon molecule pyruvate to oxaloacetate, which is a 4-carbon molecule. Okay, so that's the bicarbonate ion right here, and energy is needed. Usually, so as a substrate, you have to have ATP, and as a product, you have to have ADP. So energy is taken from this phosphate in ATP molecule. Another example is ligases that connect nucleotides to each other, forming a larger, or connecting two molecules to two DNA fragments to each other. We already talked about these enzymes in the molecular biology class. Again, energy is needed, okay, in ligating or gluing two nucleotides to each other. So another example of, so these are the 6 classes of enzymes. Now, there are other forms of enzymes that are exceptions. So remember I said that usually enzymes are protein molecules, but there are exceptions. An exception is a ribozyme. Ribozymes are enzymes that are made of both proteins and RNA. And usually these RNA molecules are the ones that act as enzymes. What proteins do is that they form the catalytic structure of the RNA molecule. So they are important for stabilizing the molecule, the enzyme itself, the RNA enzyme, rather than catalyzing the reactions themselves. Okay, so here you have an enzyme known as RNase P, and what it does, it's formed, it cleaves RNA molecules, and it's formed of both RNA as well as protein molecules. Now, usually the efficiency of these enzymes, ribozymes, is less than protein enzymes, but they do play a very important role in biology. And here's something different, okay? So we have molecules or antibodies that function as enzymes as well. So we already talked about antibodies, the structure of antibodies having a Y-shaped structure. And what these antibodies do is that they bind specifically to molecules, to antigens, foreign bodies. Now, how can antibodies function as enzymes? So basically what happens is that we take a molecule that looks like a transition state. So that's a transition state analog, a transition state of a certain reaction. So I have a substrate that is converted to a product, but in the middle, this substrate is converted to a transition state. Okay, and this transition state analog, so it looks like the transition state, is injected into mice. These mice then produce antibodies against transition states. So these antibodies have, in the variable chain, in the variable domain, they have a they have amino acids that can bind specifically to transition state analogs. And it can happen that these amino acids that are present in the variable region of antibodies, they can be similar to the amino acids that form the active site of enzymes. So when you have this interaction between amino acids with substrates, you can have the catalysis of reactions. Okay.